in December last year, Izan was tasked to put all the genomic modules together for all your breed societies, from Brahman, Simmental, Simbra, right through to Brangus, Santa Gertrudis, etc. So except for one small society, everybody now has the genomic module installed. And so tonight we're going to discuss with you what genomics is, why genomics is important, and why we've worked so hard to get a special price to be able to do the bulls and the cows, because Izan is going to explain the first part to you, and then we'll move to the other. But on behalf of the LRF and the LRF staff, warm welcome. I see we've got a really good turnout this evening. So Izan, if you can go ahead, and I'm going to allow you to share screen from my side, and if you can then go ahead and share your screen, and then start the presentation. Izan doesn't always have the best of internet. She lives out on a small farm near Cullinan, and comes into work twice a week, and that's where she's done all the work from. She's in Bloemfontein quite often. So Izan, if you can come in and share your screen, and you can start the presentation. Thank you very much, and we'll keep the chat group going. I'll see if I can manage the chat group while Izan gives the talk. A lot of you have got to know Izan very well as she travels in South Africa, and recently Namibia and Zimbabwe, where those producers are also getting to know her better. Izan, are you ready for us?
Yes, Michael, I am.
Okay. Are you able to share your screen and share the presentation?
Yes, can you see my screen?
Okay, I'm sharing my screen. There we go, Izan. I can see your screen. Let's enjoy what you've got to tell us. And of course, Meki comes in after me, but you are doing the bulk of the work. And as I say, keep the chat group going for the questions.
Michael, if I can just request you to just look at the guys in the waiting room to admit them.
100%.
I'll start with the session. Good evening, everyone. So I'm going to, now it's not allowing me to share. I have to move on.
Sometimes struggles a bit with that, Izan. You've got to just take your time with the page down.
So I'm going to go through a few topics. The first is what is genomics. The next one is the difference between microsatellites and Snips. If we can just keep everyone muted, please. Then we're going to look at the different Snip chips. What are genomic information used for? And then the DNA pipeline, and also we're going to look at the current Snip offers on the table, and also a producer perspective on the opportunity to use the special price to move over to Snips. So first off, what is genomics? So in simple terms, genomics is just the use of DNA information in a genetic evaluation. And we'll see how they do it later on. So currently we have two different scientific schools with regards to animal breeding. We have the molecular genetics side, and then we have the quantitative genetics side. So with the quantitative genetics side, this is where they use statistical models to analyze the traits. And this is the model currently used to produce EBVs for us currently. Then we have the molecular genetics side, where they actually study the structure and functions of genes at a molecular level, and they see how they can include this in the analysis. So these two schools actually developed alongside each other, and they actually started before the 1920s. But the quantitative one developed faster, and that in the 1980s, they started with the blood model that we still currently use for our EBVs. The molecular side did not develop as fast as the other one, as they first needed to develop the technologies to actually read the DNA and the markers, etc. But they started to look at the markers, etc. in the 1980s up until the 2000s, and afterwards the technology started to develop quicker, and we are now in the genomic selection era. So we're going to first off look at what is microsatellite markers. So that's the one that we currently use when sending samples to Unistel. We use this for parent verification, for detecting genetic defects, etc. So what is the microsatellite markers? These are 1 to 6 base pair repeats on an animal's DNA. So one animal will, for example, have 3 repeats of this sequence, so CA, CA, CA. The other animal will have 4, 5, etc. repeats of this specific sequence. And then they can use these markers to see, for example, what sire matches the calf, which dam matches the calf, etc., as these markers are transferred from sire to calf and from dam to calf. So they were highly studied in the 1990s, and they were the genetic markers of choice for almost 2 decades. And they are, as I said, currently still used in South Africa for parent verification. So for parent verification, they use standard 12 ISAC markers plus a few extra, up until about 90-20 markers. So they will have 12 of these. So the one will be a CA repeat, the other one will be CA, CA, TA, and then again CA, CA, TA, or whatever the case might be. And then they'll use those markers to identify the parents. So why did they move away? There's a few drawbacks with microsatellites. So they are dispersed through the genome, but there are not as many as we have Snips, and we'll look at Snips just now. And the results are not highly repeatable between labs. And this is often a problem, for example, with parent verification, where we have one lab doing the profile for the sire, and then another lab doing the profile for the calf, and often these profiles will not be compatible between these different labs, and it is quite time consuming to do the analysis. So then we move over to Snips. So what is Snips? Snips stands for single nucleotide polymorphisms. So what does that mean? That means if you think back to your biology at school, you'll remember that there's 4 base pairs that DNA are made up of. You have the A's and the T's and the C's and the G's. And those 4 are the only building blocks that we have that DNA are made of. And the sequence determines which genes they code for, etc. So at a specific position, let's say if you sequence the DNA and say start to count one position at some point and they count all the way up until hundreds of thousands of these base pairs at a specific location, let's say at location 2000, for one animal you will have, for example, a, and for the other one you will have a G. So that's a specific SNP. So they count from a specific position up until another, so they have markers, and this is a SNP. So you just have two forms of a SNP, the one or the other. And as you can imagine, you find them throughout the whole DNA sequence of an animal this is the building blocks. The ones that they actually look at is the ones where there are variation between animals. So what is the pros for SNPs? There's many more markers than with microsatellites. So you can create a very dense map of your DNA, and that allows you to have a better chance of detecting the genes that actually code for different things. They are genetically more stable. It's relatively easy to measure them, and you have lots and lots of applications that you can use them for, and we'll look at a few in a moment's time. So as I said, we have these different markers, the microsatellites and the SNPs. So you'll hear about different density SNPs. So you'll find 7K or 50K or 100K SNPs. These refer to the number of these base pairs on, or markers on the chip. So 20K will be 20,000 of these different markers. With microsatellites, you'll have markers that you use for parent verification, and you can only use them for parent verification. If you want to test for, let's say, pompies, you'll do another test, and you'll have other markers to determine those. So these are different tests for the different things you want to do. With SNPs, you run the whole profile, and with that SNP profile, you can do different things with that. You can do parent verification, you can include it in the genetic evaluation, you can detect the genetic conditions, do a poll test, for example, etc. So with the one result, you can do different things with it. And we'll talk about the compatibility between microsatellites and SNPs later on, but these are different technologies, different markers, so these are not compatible with each other. So this just shows you how a SNP chip will look. And these are just small vials where they actually extract DNA and from an animal, and they'll load the DNA within these vials. So you'll often hear that you have, for example, 96 samples on a specific chip. So there'll be 96 of these small holes. So as I said, you have these different SNPs available. The ones that we currently look at is the 50K and the 100K, and we'll talk about them in a bit more detail. So when we look at Snips chips, we often need to understand which chip is the better one or the more informative one of the two, if there are two available. A concept that I'll quickly explain is the minor allele frequency. So as I explained previously, when we have SNPs, the specific position on the DNA, one animal will have a C at that specific location, the other one will have a T. So this can be any position, so we'll call it X on the DNA or that specific SNP. One animal will have a C, that's the variant for that specific marker, and another animal will have a T. So if we look at the whole population, we'll find with the different SNPs that some of them will be more frequent than others. So in this case, if we look at the whole population, 70% of the population has the C variant at this specific position. And only 30% of the animals will have the T variant. So the frequency of the C variant is 0.7 and the frequency of the T variant is 0.3. So in this case, allele T will be the minor allele. So the minor allele frequency for this specific SNP will be 0.3. And these minor allele frequencies often contribute to the variation that we see among animals. So if we just think about it, if all the animals had a C at this specific position, then there's no variation. So this specific SNP will not contribute anything to the information that we will have on the differences between the animals. So we'll discard that. So we want those SNPs where there are differences between the animals at that specific position. So in this case, if everyone had a C, there's no T's, so the minor allele frequency for that SNP will be 0. So we want those with the higher minor allele frequencies because they give us more information. They tell us the differences between the animals. So with that in mind, we can look at the different SNP chips that we have available. So the one is a GGP 100K SNP. This is one from Neogen. The 100K tells us that there's 100,000 of these markers that we can look at. And then we have the Illumina 50K version 3. So it's 50,000 of these markers. And that's the one that is currently available for testing at Unistown. So this graph shows us the the mean or the average minor allele frequency for all the SNPs on that chip. The green bar is the GGP one, and the orange bar is the Illumina chip. So if we, for example, look at limousin in this case, the average minor allele frequency is 0.3 on the GGP for limousin cattle. On the Illumina 50K chip, it is about 0.2. So as we said previously, the higher the minor allele frequency, the more information we get out of those SNPs. So this just shows us that there's more SNPs on the GGP 100K that gives us information about the variation that we see in limousin cattle than, for example, the Illumina 50K one. But we can use the different chips as they all have common SNPs they all have common SNPs. So we can look at the overlap between the different chips. So in this case, if we look at the overlap between the 100K GGP chip and the Illumina 50K version 3 chip, there's 32,865 SNPs in common. So what that means is 32,000 of the 50,000 SNPs on the Illumina is the same as as on the 100K chip. So this allows scientists to do something we call imputation. So they can impute, for example, the 50K to the 100K just based on these 32,000 SNPs in common. So just a note that we can use the different chips. We just need to decide which one makes more sense to use, which one will give us more information for the money that we pay. But all of these can be used. Then Neogen has a Bos indicus specific 50K chip that was developed specifically for Bos indicus type cattle. This is just a comparison between the Bos indicus 50K Snip chip and the Illumina 50K. So again, we look at the average minor allele frequency, and we see, for example, for Brahman cattle on the Bos indicus chip, the mean minor allele frequency is 0.3 versus the below 0.15 on the Illumina 50K chip. So with this chip, the GGP Bos indicus chip, there will be more SNPs with valuable information for Brahman cattle than the Illumina one. This just shows a breakdown of the SNPs with a very low minor allele frequency, so between 0 and 0.04. So 0.04 will mean that only about 4% of the population will have that specific variant of the marker. So these SNPs will not give us much information about the differences between the different animals. Whereas the ones with the higher minor allele frequency they will give us more information. So they just break them into these two groups, and you'll see that in this case, 60% of the snip chip, or the SNPs on this specific chip will be in this minor allele frequency group. So there's lots and lots of highly informative SNPs on this specific chip for the Brahman cattle. But again, there's some overlap between the Bos Indicus chip and the Illumina 50K chip. There's about 11,000 SNPs in common, so they can impute from the one chip to the other chip. So both of these chips can be used. So now we're going to look at what information do we actually use these SNP profiles for. So the one is that they can include the genomic information in an animal's or a breed's genetic evaluation, which we call a single-step genomic evaluation, to calculate GEBVs. The next thing that they can use the information for is to detect genetic traits, so defects and traits like the horn-paul gene, etc. And the third thing that we can use the information for is parentage or sire verification. So we're first off going to look at how they incorporate, or what's the advantages when they incorporate genomic information in the analysis. So what is a single-step genomic evaluation? So with our current evaluations, they use the performance data for all the animals in your population and the pedigree information, and they calculate the EBVs. With a single-step genomic evaluation, they also include a third source of information, the genomic information. In a single step, they still use the BLUP, the best linear unbiased prediction model, and then they calculate genomic EBVs or GEBVs. So what's the benefits of including genomic information? So the first thing it does is it corrects the pedigrees. So incorrect pedigrees are often a problem, and studies have shown that up to 10% of parentages in pedigrees are actually incorrect. So the inclusion of genomic pedigrees will correct for these mistakes. And what it will also do is it will adjust the relationship between animals. For example, genetically, an animal might be more related to its dam or its sire. So it will adjust for these relationships, where in the current genetic evaluation, we assume that an animal will be 50% related to his sire and 50% related to his dam. So if we keep that in mind, they will check on a genomic or a genetic level to see this specific calf from this dam and this sire. Genetically, is it more related to the sire or the dam, for example, for birth weight? And then they'll see it's more related to the sire, and the EBV for birth weight will, for example, be higher than just the mid-parent value. In this case, the animal was more related to the dam, and therefore the calf's EBV is a bit lower. So then we can do the same for the next generation, and we'll see that the both these calves are from the same sire and dam. If we only had our pedigree data with our current evaluations, they will receive a mid-parent value of +9 for birth weight. But if we use genomics and genetics, we will see that this animal is more related to the sire, so they can adjust for that and increase the EBV for that animal to reflect that. And the other one might be more related to the dam for the specific trait, and they can decrease that animal's EBV a bit more, or yeah, to be a bit less than the mid-parent average. So that allows us to, with full SIPs, identify the differences between those. And this is especially valuable if you have embryos from a specific sire and dam. With our current evaluation, we will give them all the same EBV at birth or before implantation, but with genomics, we can differentiate between them for the specific traits. So what's the reason that we do not share exactly 25% with a grandparent or 50% with a parent? That's due to a process they call recombination. So when sex cells are formed, pieces of DNA are actually swapped between chromosomes, etc. What is just important to realize is that calves from the same sire and dam will not have the specific or the same genetic makeup. And we see that also in humans. You will have 4 kids and all of them differ. And we see that in humans, and we realize that, but often we forget about that in our animals. And genomics will help us to identify those genetic differences between those animals. So what's the other benefit from GEBVs? It will increase the accuracy of our EBVs generated for animals with limited performance data. So this might be animals from non-recording herds, or young animals which are not old enough to be measured for a specific trait. For example, you can have a calf at let's say 30 days of age. You haven't measured it yet for 600 days, but you want to select it for 600 days because 600 days is the trait that's important to you. So we can increase the accuracies for those 600-day weight EBVs for this young animal with GEBVs. For traits that are hard to measure, for example, feed intake, so it's expensive to measure. We do not measure all our animals, but for related animals, we can now have higher accurate EBVs for those traits as well. Traits only measured in 1/6, for example, your mature cow weights, only measured in the cows, you can have then higher accurate EBVs in your size as well, your young bulls, etc. Traits only measured once the animal is dead. So you, for example, your abattoir carcass traits, you can have higher EBV accuracies for those traits with genomics. So just to explain the concept again. So at birth, or even with the embryo, you can take a DNA sample, you can send it off to the lab to be genotyped, and then they they can calculate the GBV for that animal, and at birth or even as an embryo, you can decide if that animal is worth keeping in your herd. Will he make a contribution to your herd, or do you need to sell it off as quickly as possible? So have you ever bought a bull with good EBVs, but then after some time when you used him and you have progeny from that specific bull and they're measured for the traits, and then suddenly his EBVs change significantly? And I think if you can all raise a hand, then you'll probably all do that, because this often happens. We buy a bull, but then when we have progeny and measure those, they do not perform as we expected from his EBVs. And this is often a problem due to low accuracies, due to limited information available when you actually selected that bull. And as the progeny were measured and we included more and more information in the analysis, the EBVs changed. And this is where GBVs or genomics will make a big contribution. For young animals, we can have EBVs with high accuracies. So high accurate EBVs indicate that those EBVs will not change as significantly as we add more information on that animal's performance and the progeny performance, etc. So if we just look at an example, for a calf, if we did not have genomics, the EBV accuracy for, let's say, 400-day weight will be around 30%. If we have GBVs, we can increase that accuracy by 20% to 50%. So I can select that calf for a 400-day weight with more confidence. And the probability that that EBV will change will be less. And this is where the most benefit lies with genomics. If we have our proven bulls with lots and lots of progeny, the accuracy for them are already high, and the increase in accuracy that we will see for them when we add a genomics test will be much less. This will still be an increase, but the advantage we'll see will not be that great. So genomic testing will not improve EBV accuracies equally across all animals. So the benefit will be on the young animals and animals not measured for those traits. So ultimately, there will be an increase in the accuracy of selection. So the accuracy of a GBV will depend on the individual trait. It will depend on the accuracy before the genomics test. As I said, for those proven bulls with lots and lots of information available on them and their progeny, the increase in accuracy will be less. Also, the size of the reference population. So the bigger the reference population, the higher the increase in accuracy will be. The heritability of the trait, and also the extent of the relationship between the selected candidates, so the young animals that I want to select, and the reference population. So that is an important thing that we'll look at a bit later on again, but it's very important that we keep the reference population up to date so that the animals that we now want to select are closely related to the animals in the reference population. So with GEBVs, we can make faster genetic improvement. And we can do this by increasing the selection intensity. So that just means that we can, with more accuracy, identify those animals that are much better than the average or my herd average, etc. We can increase the accuracy of selection as we have more information available. And as I said previously, we can select those animals at an earlier age, so we can decrease the generation interval, and that will then lead to increase the genetic gain. So what will genomics or how will genomics benefit me as a breeder? So in simple terms, genomics will allow the identification of elite bulls and heifers at a young age. Therefore, breeders can make selection decisions at a younger age that is currently possible. This will shorten the generation interval, and shortening the generation interval will increase the rate of genetic improvement. So where are we at in South Africa right now with GEBVs? So for most of our breeds, we are still building a reference population. So what is a reference population? These are animals with both the genotype, so the Snip profile, and also the phenotype, or the measurement for the traits of importance, or the traits that we want to select on. So what is important with the reference population is that we want to identify all the animals based on their genetics. So we want the bad genetics, the average genetics, and the good genetics for the different traits. So it's important to realize that we cannot just genotype those good performing animals or high performing animals. We want to include all of them. So how many genotypes or animals do we need to include into the reference population? So this is a very important slide to understand. So we have our different traits with different heritabilities. So normally the fertility traits are fairly lowly heritable, and the carcass traits and the growth traits are higher heritable. So if we first off look at our higher heritable traits, let's say, for example, carcass traits that are quite high heritable, 0.5, so it's the orange graph. So at the bottom, we have the number of individuals in the reference population, and on the y-axis, we have the accuracy of the GEBVs in unphenotyped individuals. So that young calf that was just born, or the embryo with no measurements of its own yet for the trait, that's the accuracy that we will have with just his genotype. If we have a certain amount of animals in the reference populations for with both the genotype and the measurement for that specific trait. So for example, for carcass traits, if we look at the orange graph, if we want an accuracy of 60% or 0.6, we will have to have more than 4,000, around 5,000 animals in the reference population with the carcass traits measured and with the SNP profile to have an accuracy of 0.6 for that animal that was just born, or for that embryo that I want to implant. And then if we look at our fertility traits, for example, days to calving, for that same accuracy, we will need to have much more animals in the reference population. So more than 20,000 animals to have for that heifer calf at birth, have an EBV accuracy of more than 60% for days to calving, for example. So what is important here to realize is that we need to build reference populations for the different traits that we are interested in. If we want to look at fertility traits, we need to genotype cows with days to calving records. And if we want to include net feed intake in the reference population, we need to have animals with the physical measurement for net feed intake and a genotype. If we want to look at the carcass traits, we need to have animals with carcass records and a genotype. And the amount that we need depends on the specific trait, and then also the accuracy that we want to look at. But minimum, we need more than 2,000 animals in the reference population to start to look at GEBVs. So the next thing that we can use genomic information for, and that is something that we can make use of now, is to detect the genetic traits or defects. So if we have genetic defects in our population, we can snip our animals and we can identify the carriers for those specific genetic defects. And also, if we want to breed for the pole gene, we can identify the pole gene with Snips. And in most cases, it's much cheaper to do these genetic defects than when we do it with microsatellites. The third thing that we can use the information for is for parentage or sire verification. So if you think back a couple of decades ago, we used the blight types to do parent verification or sire verification on. These were actually not very accurate, but that was the technology available at the time. Then we moved over to microsatellites, and those profiles or results we could not use with the microsatellites. We had to do the animals on microsatellites if we wanted to parent verify a calf against a sire. Both of them have to have a microsatellite profile. And now we're moving over to Snips for parent verification. And again, different technologies, these are not compatible. So if I want to make use of Snips for parent verification, I need to have a snip on my calf and a snip on my bull. So the International Society for Animal Genetics, ISAC, supported the transition from microsatellites to Snips for parent verification, and they recommend 200 markers to be used for parent verification. If we compare that to the 12 to 19 markers used when we do it with microsatellites. You can imagine that the 200 markers will be much more accurate, and we can more easily distinguish between closely related bulls. So if we use two brothers in the same herd, we often struggle to identify the bull or to exclude the one or the other. With Snips, we can easily do that as we have access to more markers. So again, just to emphasize that, if we want to move over to Snips for parent-feed testing, we need to have both the calf and the sire and the dam on Snip profiles as well. So the next question is, can I stop recording the phenotypes of my animals once my breed moves to GEBBs? Can I then just take a hair sample, send that off to the laboratory, and don't need to do anything else on my farm? The answer is no. There's need for continuous phenotyping and genotyping. As I said previously, for genomic predictions to work best, the young animals that I want to select now must be closely related to the reference population. So I need to add animals to the reference population on a continuous basis. So I still need to phenotype my animals, measure my animals, and keep genotyping as well. So then we move over to the DNA pipeline that we implemented for the societies. So this is just simply the breeder sends his DNA samples to the laboratory. He fills in a form. The society sends it off to the laboratory. The results is sent back to the society, and the producer receives a report back from the society. So what's the advantages? It takes some of the administrative burden out of DNA testing for the breeder. It reduces the possible duplication of animal IDs between the societies and the laboratory. So a breeder might send a different animal ID or with a dash or a slash to the laboratory, and it's then different from the animal ID at the society. So there's sometimes duplication between the animals, and that can be eliminated. It facilitates the registration process as the results come back to the society. If we send samples from the society to the laboratory, we have a collective bargaining ability with regards to price, as we now send 1,000 or 2,000 samples and not just 10 or 20. Faster turnaround times due to less mistakes. As the society check the samples, they check the IDs on the samples, etc. And the samples can also now be stored at the society, so they can build a biobank for future use. And then also the results are sent back directly to the society office. So it will not be your responsibility to send in the parent verification results to the society to get the animals registered. So what does the breeder need to do? You will need to send in the sample to the society. This can be hair samples or tissue samples. If we look at hair samples, we have a Newgen hair card that you'll hear about. So what is the advantages of using hair cards compared to normal envelopes? There's less contamination as you stick the roots there and you cut the excess hair. Then at the laboratory, they just punch a hole in this and punch that roots in a test tube. So they do not actually physically handle the hair sample. So there's less chances of contamination. And it's also easy and quicker for the laboratory to use. So there's faster turnaround times. And there's also barcodes on the hair card that they can actually trace. And at the laboratory, they scan that barcode. So there's less chances of IDs that they type in incorrectly, etc. And they are quite small, so there's little storage space needed. So the next thing that you need to do as a breeder is to complete your 780 form. So this is an Excel spreadsheet similar to your registration form. So you simply fill in the animal ID. You say which sample you are sending. Is it a hair sample? Is it a tissue sample, etc.? To which laboratory do you want to send the samples? And then you indicate what test do you want this animal to be tested for? Is it just a SNP profile, or do you want to include some of the genetic defects? And then you have the different codes for the different tests. And if you want to do PV, you say Y for yes, and you give the sire ID, etc. So it's quite simple to do. At the society, they just pull this into their system. An automatic request form is generated for the laboratory, and at the laboratory, they can electronically pull that again into their system. Then the results are sent back from the laboratory in an electronic format to the society. They pull it into their system, and a report similar to this one will be generated. So you will have the animal ID, name, etc. And then you have the test that this animal was tested for, and it gives you the result every year. So it says here the animal has a genotype profile, a GGP 50K, tropical beef, that's the Os indicus strip. And it was also tested for CMS and Pompies. It's free for CMS, free for Pompies 13, but a carrier for Pompies 7. And then you'll have a report like this for all your different animals. So if we look at this carrier, I looked this bull up on the system, and he has 34 calves registered. 3 of them were actually recorded as aborted. 3 of them were recorded as died before 48 hours after birth. 2 of them died 48 hours after birth, and another 2 were registered and they were indicated as died. So this is probably due to the effect of Pompies. So if this breeder had tested the bull before it was actually used, he could have had 10 more calves from this specific bull. This is just another example, a Wagyu example, where we have all the defects that they carry. For this animal, it was free for all of the defects except for F11. there's another F11 carrier, and there's another one. So Snips easily gives you the results on all of these genetic defects that might affect the efficiency of your bird. So just quickly, the things to remember about genomics. It's not a magic tool. Accurate phenotypes still need to be collected. The concept of garbage in, garbage out still apply to genomics. And genomics is not an exact science, but currently it's the powerful new tool that we have for predicting EBVs. So then we can move over to Michael with the different offers that we have currently available for SNP testing.
There we go. Thank you very much, Izan. That was a lot of information for our producers to take in. Whenever you hear new information, the new words, SNPs, you'll hear MIPs. Tonight it will all be like new. But as you work with the stuff, we have our technical committee and the various societies, breed improvement committees working on it. And then within 5 months, you know all the terms. And then you start arguing and debating which SNP and which the cost relative to the SNPs and all these kind of stuff. So yes, you're going to need to hang in there. But as I said on the chat group there, testing for a SNP is again a once-off thing on an animal. You do it once, but you've got to then do the sire of that animal. You've got to do the sire of that animal. And so you move across from one technology to another technology. And so we went to the various laboratories that are available. And at times, one laboratory will give a significant special price. So one laboratory approached us and did say they're prepared to give us a special price. The board requested that we open the offer to other laboratories as well. So the Unistel offer was that we do at least 1,500 samples, and the cost would be 650 Rand per sample for a SNP test on a bull or a cow, and that will also include the genetic defects. We then went, we can move on one, Izan. Izan, are you still there?
Yes, sorry, Michael, it again doesn't want to move over, so let me just share my screen again.
Okay, good.
Apologies for that.
We then went, another laboratory approached us, Neogen, which is easily the world's largest genomics company in the beef industry. And they said, well, to help you move the technology from microsatellites to SNPs for the parental animals, the sire, and if you want to, the dam, also your influential animals, because that's what you want to do when building a reference population, and your animals tested for difficult to measure traits. Meki will talk about that now. And meat quality, if you do it on all those animals, not the young calves being born, we will provide it for you at a cost of 370 Rand per sample. That will include the genetic defect, that price, but it'll exclude the Neogen hair card cost. And Neogen is only prepared to accept DNA vials or hair cards in their laboratories. So that's the offer on the table from them. And the samples must be submitted before the 1st of March, but it'll be sometime in March they'll say, okay, now you guys in South Africa need to consider cutting off on the special offer that's available. If you can go on one. And some of the societies have also decided, Meki will discuss that, to actually subsidize that cost so that we can do influential bulls, etc. And you can speak to any American or Australian, and they will tell you that this is a very good offer on the table. So after March, we go back to the normal cost structure, which is in Neogen's case, 620 Rand per sample. That includes a fee that goes to the society for 25 Rand for administration, but that excludes the genetic defects, and each defect then normally costs about 20 Rand. It comes at an additional cost, but we have put together bundles for you. And it also excludes the hair card cost, which your society will determine the price. You can go on, Izan, or am I finished now?
You're done now, Michael.
Okay.
Just to highlight again, so this offer of 370 Rand is not for calves. So for those calves, you want to test them now, you will still need to pay the 620 Rand.
That's correct, Izan. So if you can, and we can please put up your hands. Let's discuss these things. It's a new technology, building a reference population. What do you have to do, etc. Mickey will cover some of that as a producer, but please put up your hands and let's ask the relevant questions. Izan, have you got Mickey available?
Okay, Izan, it's Mickey here. If you could give me the sharing of the screen, please, Izan.
Mickey, you can share. Mickey, we can see your screen now, so you can start.
I just want to enlarge it.
Sometimes takes a short while and you've got to just be patient.
Okay, thank you very much, Izan, and good evening, everybody. I've been asked to make a contribution here just on as a producer who uses this technology, and I always compare this with the driving a car. I've got full confidence that there are engineers and, in this case, scientists, geneticists who do the job. In Armidale, there are, I think, about 22 scientists who do this. So I'm full of confidence that they can do their job, and I just drive the car. In this case, I just use this technology as a breeder, as a producer, to do better breeding. So let me just then start off. I always come back to this one thing. Why do we do all this? And I just want to leave you with a global perspective, because we are in the beef business and we are supplying proteins to the world. So the challenge in a couple of years' time will be that we have to double the feed and we have to double the food for 10 billion people. And it's only technology which will assist to achieve that goal. And we've seen that in all facets of development. Of course, there is one important focus that it must be done on a sustainable basis, as we have done, as you see now the last week with climate change. It must be done, technology must be used sustainably. So for the livestock sector, which provides a central source of protein for the world, one application of technology here is genetic improvement, and the new technology of genomics plays a very important role here. So we as farmers, producers, we we always come across these new challenges, and it's the one, it's not the fittest one which will survive, but it's the one that is most adaptable to change. And we as producers and farmers must embrace that we take up these new challenges and that we are ready to adapt to this new technology. So I believe genomics is a next big step in animal breeding. If you look at the history back, about a century ago, it was visual appraisal. Then the pedigree assessment played an important role in selection. And about 4 or 5 decades ago, we had the introduction of BLUP, which then led to estimated breeding values. And in the last 30 years, performance recording became a dominant factor. And for all successful breed societies internationally, and that resulted then in the use of EBVs, as we have known this for the last couple of years. So the next step in animal breeding is the implementation of genomic information in the selection process. And this is where the LRF has come in at an early stage already, about 18 months ago. The LRF, together with Breedplan, has engaged in creating genomic modules next to the pedigree module and next to the performance data module. We implemented for all LRF breeds which participated the genomics module, and that was done on a more collective basis, and for that we could get a better price. So for me as a producer, the advantages are the following, and I think Izana set the scene here. We as breeders in the different breed societies, we should invest collectively, all of us, to build this reference population. It's of very little use if only 1 or 2 breeders do this. The reference population, as Izana has shown, must come from the whole range of genetics available for this breed, from the bad ones to the good ones, and from preferably from each breeder, because that will lead to a good reference population. And with that good reference population in place, and of course with the phenotypes, we will move to GEBVs, which is a much more accurate way of expressing breeding values. So for me, the one important thing for the genotyping and GBVs is its high accuracy for difficult to measure traits, and thus a faster selection. And I've listed a couple of traits for myself here. The first one is fertility. There are certain aspects there which we are not yet at, but I think in 10 years' time we will address them in terms of semen quality, puberty, and estrus, etc. And if we can collect the genotypes now already, that will be a very critical one. The second one is gestation length, is mostly measured through AI. Artificial insemination is an expensive process. So if we can have good information there, then we will get good EBVs for gestation length, and thereby shorten the production cycle. Milk is another one, difficult to measure. Carcass traits, we do ultrasound scanning. The LRF has bought a machine, it will be used in the coming year. With that ultrasound scanning, we measure eye muscle area, rib drum fat, intermuscular fat, etc. But it is an effort. You have to go out, the scanner has to go out to different breeders, to their farms, to the property. You spend the whole day there in the kraal measuring this. So it's a lot of effort measuring carcass traits. The next one, which is difficult to measure, is feed efficiency, which we believe is one of the most important traits which we will be confronted with in the decades to come. We as in the livestock sector are far behind the other protein sources in terms of feed efficiency. If you look at pork and if you look at poultry, much more efficient, and that's what they've done the last decades. They focus on feed efficiency, and we have only now, as livestock breeders come on board here, it will be a very important trait, difficult to measure, very costly. It's about 6,000, it's a 4,000 to 6,000 rand a test per animal over a period of about 70 days, plus a 21-day adaptation period. So it's a long period and it's an expensive test. The last one, which I consider the difficult to measure traits, which I will focus on, is meat quality, and then in particular the tenderness. All the other stuff like marbling, etc., we leave for other breeds. They're better with that, but I think the issue of tenderness is an issue which we have to address, because we are in the beef business. To carry on, genotyping and genomics will allow me to do much earlier selection. Izan has mentioned that it can be done with born calves or even with embryos. If it's worthwhile, carry on with that calf in the direction which you want to move. The next point is, for me and for any breeds, is the defects, which Izan has also talked about. For Brahman specific, it's the CMS and Pompies, which are the two most important ones which you look at, and we estimate that about 2% of calf loss per breeding season could be avoided if we pay attention to those defects. And each breed has got its defects, and you should look at it and try to rectify it. So in this particular offer, which Michael has talked about with Neogen, for that specific price, all the defects are included. It will give us a big boost, and it will, I will definitely participate to make this, identify those carriers which are disadvantageous for optimal production. We have said that the pedigree will be much more accurate. It is estimated that in general now, for all breeds, that there's an error of about 10% in somewhere in the pedigree. That has been scientifically proven. So parent verification is very important, and the genotyping animals, of course, helps in finding the correct parent. The LRF, it's specific, has implemented a parent discovery module that allows the program to find, to search for the sire of an animal that is unknown. And this, of course, needs to have a large population of snipped animals if you want to do that, because all the sires must be snipped. Most societies, or many of our societies, have now started to make it compulsory that all sires are genotyped. I think this is an excellent move, because by doing that, you have covered already half of the pedigree, and you have got a much better basis to find those animals if they are unknown. So on the stud breeding level, what we have done here as producers, we participated through the LRF in the Beef Genomics Program 1 that was concluded about 2 or 3 years ago. The main focus of that Beef Genomics program, and it was available for all Southern African breeds. There's an organization with the name of TIA. They invested about 36 million over a 3-year period. Those breeds in South Africa through the LRF, which participated there, they gained a lot of, or got a lot of funds for doing that. And it was I think that the program was assessed. It was found very productive and a good return on the investment. And we all hope that Beef Genomics Program 2 will be starting now. There was a bit of a hold-up with the funding, I think mainly due to COVID. But let's see if, according to Professor Mwashe, that should be on the cards again in the coming year. But there we focused on difficult to measure traits, especially NFI, meat quality, and then of course also genotyping. In the Beef Genomics Program 1, all stud sires were genotyped free of charge. So those breeders which participated, they got a free genotyping of sires done by the ARC then, which means half of the pedigree was done. Now, as a Brahman breeder, I can only talk about Brahmans. We have in Southern African Brahmans, that includes all Southern Africa, and Zimbabwe is part of our monthly run since about a couple of months ago. As far as genotypes is concerned, we have about 900 genotypes ready. You remember Izan's graph, this is not much. So our goal is to achieve as soon as possible 200 genotypes as fast as possible at lowest cost. And this offer from Neogen is an excellent opportunity to engage and to do that as fast as possible. We are using the 50K Indicus chip, which was recommended already some time ago by Professor Hayes. He's one of the world experts in genomics. And Izan has shown you the differences there earlier today. As far as we are concerned, in our stud, when we heard about Neogen, we identified some of our best 31 cows. We genotyped them together with the defects, and we got some feedback, and one or two had defects which we addressed. So this was also used to ensure that the pipeline from Namibia works. Other breeders did the same in South Africa, and this was done at own cost, not at the special price. But for us, it is important to engage as early as possible and to make use of that new technology. Together with the Namibian Stud Breeders Association, by name Mr. Jacques Els, we structured the genotyping process in order to be as effective as possible with low cost. So it's little use to genotype randomly. You have to genotype animals, and Izan has stressed this, with, if possible, with complete phenotypic data. So select those animals which are influential. The sires are one, and then of course all the females. And I don't know if you could read this, but under the Southern African Brahman Breeding Forum, is together between Namibia and South Africa, the breed societies has decided to fund this project of genotyping together with about 300,000 per annum for the next 3 years. And Namibia had made provision for another 100,000. So together, Southern African Brahmins, we have budgeted to invest 1 million in genomics in the next 3 years, and we hope we can do it as cost effective as possible, because we see here a big future, a bright future, and good results. The price which Michael has offered now or stated now it will start in, well, it has started already actually. I know certain breed societies have sent off data already, or samples already to Scotland to do the genotyping. I personally, we looked at all our old stud sires, which we had DNA samples stored here over the last weekend. And we enjoyed our treasure box, which we had. And these are old sires with high accuracy EBVs up in the far 90s, 96, 98 for growth, for example. A lot of progeny, 100, 200, 300 progeny sometimes. These are good old sires which you can use to build up the reference population and make a contribution. Then we also identified a range of young sires, and I would recommend every breeder listening here tonight that you go out and use all your sires you've got, pull the hair, send it to the breed societies. We've done all the sires up to about 2 years ago. We will do the last one. Those which we used the last 2 years, we will definitely, we've actually already started pulling those hairs, and we are ready to get them on the way to Scotland. We will, in the next week or so, we will go back to our differential females and pull hair samples to get them genotyped. That is an important action, and we will also identify those animals which we have tested for difficult to measure traits. And the first one is net feed intake tested bulls. We will have to make a selection there of bulls which were used. All bulls which were tested up to now for net feed intake have samples stored. And the same, if you look at the other difficult to measure trait, that is meat tenderness. Not so many bulls as for net feed intake, but we have to start, and those samples are also stored, and we will also make use of those samples to get them genotyped. And then, of course, most sires which we have, ultrasound scanning was carried out, so that's the easy one. Genetic defects, I've mentioned. We will test all animals at that cheap price, free of charge, for all the genetic defects. So let me just come back. These are my last 2 or 3 slides. On the issue of net feed intake, we have many bulls, and they can make a big contribution to the genomic reference population. And once that is done, all breeders will profit from that, because if you send your hair sample in from your stud sires, you might get an indication of a net feed intake value for your stud sires. These animals have both been phenotyped and genotyped, and they can become part of the reference population. And we have seen now that with the EBVs for net feed intake, which Brahmans have now since March 2020, many of the old animals in different herds, they have got EBVs for net feed intake and quite high accuracy, because their progeny was actually tested, and they can also make a contribution to the reference population. At our net feed intake test station in Namibia, Gentex Sol, we are busy now for more than 5 years now. We have tested 1,300 young Brahman bulls from different breeders. In total, we have tested about 1,800 young bulls for all breeds. All these 1,800 bulls hair samples were taken, they are stored, and they can make a big contribution to the reference population. I just want to show you these two pictures here. This is just the indexation of genetic soil. Those who are not so familiar, on the left-hand side are the feed bunks where the exact feed is measured, and on the right-hand side are the water troughs where the partial weighing of that animal is made on a continuous basis while it drinks water, and it will give you a good indication of the weight and thus the weight gain. The other topic is meat tenderness for Brahmans. In BGP1, we have done this, we've tested 130 different Brahmans from different breeders in Southern Africa. And the shear force is the vertical axis here. And you can see the green and the blue and the red bars here. Brahmans have got genetics which are restaurant quality, and in our case, where we already in the 90s with AI experimented with that, we could add it in the first 10 here, in the first 10 green ones, 6 are from our stud, which means that you can select easily and you can focus your attention on that. In South Africa, the Brahmans have done a big new test. They're still carrying on with that, with good new data. So meat tenderness is another one of those difficult to measure traits because you have to slaughter the animal, and it's if we can improve that trait by genotyping, then that will be a big asset. So from my side, we will definitely heavily engage in genotyping, and I trust that all other breeders who are listening tonight, who will get a focus on their future production, will join this train and don't miss it. That's all from my side, Michael and Izan. So thank you very much, and I will stop sharing my screen.
Thank you. Thank you very much, Mickey. I think there are a few things that came out of tonight's slides. One of them is what do you use genomics for? The second one is how do you build a reference population? For us as societies, that's we say in Afrikaans that is a kopkrap oefening, because you want value for money. Genotyping is expensive, so how can you do it in a cost-effective manner? And that's why Izan looked at those 3 different reasons for going to Snips. One is accurate parentage, but you can argue that you can already do accurate parentage on a microsatellite. But the second reason becomes accurately doing genetic defects. Then the pendulum starts swinging in the favor of Snips and genomic values that come with it. And then the third one is when you can create that big enough reference population, because you're already building the DNA samples on the parentage, etc. You can build that big enough reference population to turn on the machine to then give you the genomic breeding values. In Mickey's case, for example, where he will use it is Mickey sends about 400 or 500 of his own bulls to the feed efficiency station. He will now be able to go and pull hair samples on the calf when it's born, and then decide based on the result of the genomic EBV whether this calf is worth testing in the feed station or not. So you'll probably find a third of them will not make the grade, and then the cost benefit swings completely in the direction of genomic Snips. It's done it to the extent that no more microsatellites are done in Australia for any breed society. The same with the UK. And when you speak to Neogen America, they say we reluctantly do microsatellites. What we do in South Africa, we reluctantly do microsatellites on an animal because we realize you're losing all that genomic information that you could have had on that progeny. For breeds such as Wagyu, for example, where every animal is carcass tested, if you don't do genomics on that animal, that information, and you just do a normal microsatellite parentage test, that information is forever not available on that, how that bull would have bred, where you could have gone and got an early indicator of that bull's breeding. Are there any questions? I know this has been a long talk. We apologize for that, but Mickey, it's the first time I think we've got 75 people on one of our LRF talks. We normally have 30, 35 when he's with Izan doing a lot of the modular courses, etc. Are there any questions? Because often one question sparks a few questions, and if you can ask a question, then somebody else also understands a little bit better. So any questions? Any questions from any of you attending? Okay, so I don't see any questions. The recording will be made available on our website, but you can contact Izan and myself anytime and ask questions. We have been engaging very strongly over the past year with your breed improvement committees and also society AGMs, etc., where we're explaining this technology, explaining why it's important. We didn't even talk about RLR Online, that in this last month for all the societies we've been putting in place, where you can go into the database and basically do exactly what the staff member at the society can see and can do with some check systems built in there. It's already been set up with 3 or 4 of the societies and working very well. So I just want to look, there's a question here: for the purpose of the special offer, what is a calf? What age measurements is on? You need to come in here. We've been talking to Neogen about this. I think they're not hulle gaan nie hare kloof Dewald. But if you send in a month-old animal, they're going to say this is a calf. But if you send in a year-old animal, you can argue that this animal, or 18-month-old animal, you can argue that this becomes a heifer, and that's why you can want to do this young heifer, for example. So I guess the definition, if we're going to make it up ourselves, a calf is an animal that doesn't yet have pair, or a calf is an animal that is below, that is not himself or herself reproductive yet. That was Dewald's questions. Any more? I like questions like these because that's where we really learn the most. I see Bianca, you from Namibia, I assume, Bianca Brown. Is daar iets van jou kant af, Bianca? Bianca is also slightly shy.
Yeah, good evening, Michael. Yes, maybe also in that line, the bulls that are going to the NFI tests are also very young. Can we send those samples?
Absolutely, Bianca, because those are the perfect reference population, because you now got the genotype on the bull, and you got the phenotype, the raw data of that bull in a test for a difficult to measure trait, so that you can go in future and when that bull is a calf, you pull a hair sample. These genomic tests will always come down. They're like laptops and things like that. So let's say they come down to 400 Rand or 300 Rand in the next 4 or 5 years. You can then go and you test that calf and you say it's not worth sending this calf to the test station. And that's exactly what the reference population information is. Same with the heifers, with young calves, or old, not heifers as much, but old calves with reproduction information. Ideal for building up a reference population. Does that make sense?
Okay, thank you, Michael. And yeah, it makes perfect sense. The question is now, because say the bulls that are going in or are at the station now, will they qualify for the special rate because they are so young?
Yes, put them in. We can argue on your behalf that we want to build this reference population, and that's what we're busy doing. And they've encouraged us to do that, to say, see how quickly you can build your reference population. Look, there's money in it for the genomic companies as well. Make no mistake. Once we've all moved across to genomics, the price goes back to the normal 620 Rand. But as I said on the chat group here, you're then getting lots of information for that amount of money. Whereas if you do a microsatellite at this moment, you got the parent result, but that's all you've got. You don't have any genomic information on that animal. So the Americans will tell you it's a waste of, you've wasted the 150 or 170 Rand, whatever you pay for just a parentage test.
Michael, there's
Okay, thank you, Michael.
There's another.
Then somebody asked a question through Snips. Will the genetic defects be displayed on the bull certificate? That's for your society to decide. But what we have decided as LRF societies is to put all the information, bulls and cows, etc., onto the Internet Solutions website. So you'll be able to look up a bull and see if it's a carrier for a trait or not. This is accurate. These are accurate results. We gave a small, short farmers talk about a month ago. Elandri, I see she's on there. She was with me, and Izan as well. And this farmer had quite a lot of bulls with AIRS. He's a Wagyu breeder, AIRS, which is a lethal genetic defect. And we could show him on his form. So you got a carrier, an affected animal. And we said, this young bull died. And he said, how do you tell that? And we showed him on the form, and he got out his little book and he paged through his book. And sure enough, a year ago, that calf had died, which was a lethal carrier. So we don't want to make too much of the fact that now we can instantly expose which animals have genetic defects or not. But it is one of the advantages of doing genomics the way genomics is done. And if you go onto any Australian website who also uses BreedPan, you'll find those defects listed for every single breed. Yeah, so that certificate, that'll be up to your society. Would it be useful to move on this technology if our respective societies are not? This is a society type of initiative. Because if you move on your own, you're never going to be able to build the reference population that you require to really get the benefit out of genomics, which is a genomic evaluation. And if you start today, you'll only get your genomic evaluation in 2-3 years time. It's a long process, but you've got to get to those 3,000 records. Brahman are very close, and so that's why they got a big push. Wagyu have also climbed on the bandwagon, as Simbra, for example. But the other societies really need to take this on board, and we're there to assist you. Izan has sat with every society and help them with a pipeline so that we can get those pipelines going. So it's better to do it in a society. That's a good question. Dewald, I see you. Jy nog 'n vraag, Dewald? Nice to see you as well, George Gibbons. I see you don't have your volume on, but we really need to work with Brangus as well to get this pipeline going. If you look at the USA Brangus, they've also, I think, a third of all animals in their data in their pedigree databases are genotyped, if I remember correctly. And they've also employed a CEO to basically move them into the genomic, into moving the genomic era. Okay, Mickey, I think I don't see any more questions. We'll have another one of these sessions, thinking about February, Mickey, because that will be before March. And by then, I think there might be a lot more questions, because a lot more producers will be wanting to find out about the technology, but a lot of producers will also be already testing for the technology. So on behalf of Mickey, our chairman, and Izan baie dankie vir jou. And Izan is a Namibian girl. This is the longest I've heard her talk English. Baie dankie, Izan, dat jy so vlot lekker Engels gepraat het. En ek dink vir 'n uur, as ek reg onthou. Ons waardeer baie. Okay, thank you very much. This will be recorded, and we'll see you in February sometime. Thanks a lot.
Thank you. Bye-bye. Thank you.